Okay, thanks for that introduction, and I'd like to thank the Institute of Medicine for inviting me to participate in this session. I took a look at the description of me in the program, and I was intrigued to see that I'm the author of a book on aquatic pollution that's been translated into numerous languages. It's true that I'm the author of a book on aquatic pollution. It's been translated into 2 languages. So I infer that here in Washington, any number bigger than 1 is numerous. Now, the subject I'm going to be talking about today is, unfortunately, a very imperfect state of affairs. It's monitoring of recreational waters, basically to determine whether it's safe to go into water or not. There's actually a lawsuit involving the EPA on this, which is supposedly going to be resolved at the end of this month. It'd be very interesting to see how that plays out. But, as I go through this, you'll see there are a lot of issues, and some of them are in a state of being resolved, and some of them, frankly, it's not clear how we're going to deal with them. This is a paper that was published back in 1997, just to point out the value of recreational waters. Bob Costanza, the senior author on this, and I mean, in this paper, the authors looked at a lot of different types of ecosystems, one of which was recreational waters, and tried to put a dollar amount on it because that seems to get the attention of people in Washington. So here's a summary of various ecosystems and their values, and you can see these are billions of dollars, and this is global. Nutrient cycling was perceived as being far and away the biggest ecosystem service provided, and then you can see as you go down the list, and finally you get down to recreation. It's in 9th place, and there are 8 other services lower on the totem pole, so it's right smack in the middle of the pack. It's not the most important one, but it's certainly there on the list, and $815 billion per year in value. When you look at the breakdown of this, you see that most of these ecosystems are aquatic. Not all of them, but a lot of them are aquatic. You can leverage this a bit, and so here are some students. In other words, you can leverage, in this case, education and recreation. These are students from Kamehameha Schools, which is a school in Hawaii for students, for children of Hawaiian ancestry. And in this case, the program was organized as a part of this Center for Oceans and Human Health at the University of Hawaii. So the idea was to mix education and recreation. I'm not sure how much, what the percentages are on this, but you can see they're looking at a sample of water there in an interested sort of way. Now, this is taken off the internet, and it's some verbiage that relates to this ongoing lawsuit, which I've highlighted here in red. It's a responsibility, EPA, to set these water quality, establish water quality criteria. It's actually up to the states to implement them. But there has been, to say the least, dissatisfaction with the water quality standards that have been promulgated by the EPA, and this eventually resulted in a lawsuit, as you can see. It was originally scheduled to be settled prior to this meeting in October 2012, but if you read down here to the bottom, you can see that it's been extended to November 30. So in a couple of weeks, we will presumably find out what the resolution of this is. I mean, actually, we have a pretty good idea what the resolution is going to be, but you can see it's been contentious. It's gone to court. And no matter what the EPA does here, frankly, they're going to continue to be ongoing issues, frankly, for quite some time. So if you go back to 1972, when the EPA first started to deal with this issue, there was remarkably little information on which to base water quality criteria. In fact, you're looking at it. They had some information on counts of so-called fecal coliform bacteria, and you can see the range of the counts in the water. And then they had just taken samples of these waters and assay them to see if salmonella was present. And so if you had somewhere between 1 and 200 fecal coliforms per 100 mils, 32% of the samples contained salmonella. And the higher the number of fecal coliform counts, the greater the percentage of the water samples that contained salmonella. That was it. And so it was very obvious to the EPA that something needed to be done. And so the EPA organized some what they call epidemiological studies. First of all, they had to find recreational waters in the United States that were polluted with fecal pollution. And these had to be waters that people were actually going bathing in. Well, you might ask, well, wait a minute, if they're contaminated with human feces, why are people going swimming there? Good question. And it was frankly not easy to find these places, but they were able to identify some. And they had to do both marine and fresh waters. And the idea was to assay these waters for a laundry list of potential so-called indicator organisms that you would associate with human feces, and then correlate that with the incidence of people getting sick from going into the water. And going into the water to the EPA meant you got your head underwater. So if you just waded in, that didn't count. You had to put your head underwater, but it didn't matter whether you did that once or numerous times or how much water you swallowed. And I just mention that because if you go to a place like United Kingdom, there's very different attitudes about how to go about this. And so in the UK, for example, they force you to go in the water and stay in the water for a specified period of time, and you have to do various things while you're in the water. You can't just stand there. So there are different points of view on this. And then during the roughly week or 10 days after these folks went in the water, EPA would follow up and ask them, did you get sick? And these had to be illnesses that you would associate with ingesting pathogens related to feces. So if you were throwing up, that counted. If you had diarrhea, that counted. But then, watery stools. So what is diarrhea? And so these had to be what they call highly credible gastrointestinal illnesses. And then they correlate that with the concentration of these various indicator organisms to find out if anything correlates with the occurrence of people getting sick. And this is a summary of some of the results that they obtained. Now, these are in marine waters, and these are Enterococcus bacteria. They're found in everybody's gut. It's nothing to do with whether you're healthy or sick. So when you defecate, you shed these things, and so if there have been feces in the water, you would expect to find Enterococcus. And you see, here's the concentrations. It's a log scale, and here is highly credible gastrointestinal illnesses, per 1,000 swimmers. Now there's a correlation, and now that one turns out to be statistically significant. Another organism that they looked at, E. coli, which again is found in everybody's gastrointestinal system, and they went ahead and did the regression, but if you look at the data here, you can see they're more scattered, and statistically that relationship is not significant. So it turned out, although they went through a long list of potential indicators, in marine waters this is the only one that correlated with people actually getting sick. It's Enterococcus. Now when they went to fresh water, they found that both E. coli and Enterococcus correlated significantly with people getting sick. And so the water quality criteria, which are still in place, at least till the end of this month, for fresh water, you can take your pick. You can use Enterococcus or you can use E. coli. If it's marine waters, you have to use Enterococcus. And so these criteria then came out in this famous or infamous so-called Gold Book, 1986. And this concerns all sorts of water quality criteria, but among them is the occurrence of fecal indicator bacteria. And these are what the standards are. The geometric mean, and it's specified in the fine print, how many samples you have to take and over what time frame and so forth. And see, this is fresh water, so you can use E. coli, you can use Enterococcus. And there was a lot of discussion about how many people is okay to get sick, because it's a probability thing, right? And they put this out for public comment, and you can imagine they got a lot of feedback on that. So these are the geometric mean criteria. And then if you just take a single sample, which frankly is more often the case, these are the criteria. If you go to marine waters, the geometric mean, again, see, it's just Enterococci here, 35 per 100 mils. If you actually take the samples, enough samples over the time frame that they specify, the geometric mean has to be less than 35. Take a single sample, it's 104. Now I want you to remember that number, because I'll come back to that as we look at some of the other slides. A single sample has to be less than 104 Enterococci per 100 mils. Okay, so what are the problems with going about things this way? First of all, the organisms we're looking at are not the pathogens, right? They're just indicators. It makes sense because, I always say they go down to the water and they take a sample about the size of a teacup. And so you want the indicator to be very abundant in feces so that when you take your teacup full of water, you have a good chance of detecting the presence of the indicator, because if you're talking about the actual pathogens, hopefully the water's not full of them. And there are a variety of these pathogens. There are bacteria, and I'm sure you're familiar with some of these. This is not an exhaustive list, okay? It's just some of the ones that more often than not turn out to be problems in the United States. You have viruses. And again, this is by no means an exhaustive list, but some of them. You have protozoans, and I'll get into this in a bit. These turn out to be problematic because, at least in some cases, they are not removed by standard ways, for example, by chlorination. No, it doesn't take these things out. And then you have worms. Schistosoma is the principal one in the United States. If you go to other parts of the world, you have tapeworms and things like that. And then I want to point out that 70% of hospitalizations for gastroenteritis in the U.S. are due to unknown causes. And in fact, when the EPA did this study, they have no idea why these people were getting sick. They were vomiting, they were having diarrhea, stomach cramps, but no idea what the causative agent was. And it's expensive to assay for all these things, to say the least, and particularly in the case of viruses. It's not simple. And the departments of health do not have the budgets to go out and hire a bunch of PhDs to do all these assays. So they're trying to come up with something that's simple and fast, and that's why they use indicator bacteria. But with the realization that the fact, there are a wide variety of organisms that may be causing these people to get sick. And you're trying to use a single indicator to determine whether these things are present or not.
Is that 70% number back in 1986, or is that today?
It's today, yeah. No, I mean, people come in, they're sick. If you want to know what caused them to get sick, you got to take a sample of their stools and go through and look for all of these things. And a lot of times it's not done. So here's a couple of examples where the system failed. This is Delhi, India, 1955, 1956. The public water supply became contaminated with sewage. It was during the monsoons. And the authorities ramped up the chlorine, and they assayed the treated water by looking for fecal indicator organisms, in this case E. coli. And they had standards there, and the water met their criteria. It was safe to drink. Well, you can see that there were 20,000 cases of infectious hepatitis. And the point is that viruses in general are more resistant to chlorine than, let's say, bacteria. And for whatever reason, the infectious hepatitis or hepatitis A virus is particularly resistant. And interestingly, there was no increase in the incidence of any other kind of infection other than infectious hepatitis. So apparently they knocked out every other pathogen in that water except for that one virus. And they had 20,000 cases of people getting sick because you're relying on E. coli, which is very susceptible to chlorine. It's a bacterium. I'm sure they completely wiped it out, okay? But they did not take care of the infectious hepatitis. And not to be outdone, here's the United States, about 40 years later, Milwaukee. 400,000 people became ill, and this is a protozoan, Cryptosporidium parvum. It is absolutely not taken out by chlorine because it forms in cysted eggs. They're resistant to chlorine. You want to remove this thing, you have to filter the water. And they were giving it standard treatment. They're looking for fecal indicator bacteria. Water looks fine. You have 400,000 cases of people becoming ill. So there's an example where reliance on fecal indicator bacteria can completely mislead you, because here you have two pathogens that can completely escape the wastewater treatment process or the water treatment process and obviously make a lot of people sick. Then you have this problem, and it takes 24 hours by the traditional tests, and this is one of the issues the EPA is wrestling with in this lawsuit. It takes 24 hours because you've got to grow these things on a specified medium and count the colonies at the end of 24 hours. So this is a study done by Allie Beam at Stanford, and she went to this recreational beach, and she's looking at the correlation between Enterococcus counts. So these are marine waters, so you have to use Enterococcus. Enterococcus counts measured on one day versus Enterococcus counts measured on the next day. Keeping in mind it's going to take 24 hours to get the results. And you can see the correlation, and there's no correlation. So, the story that Al Dufer from the EPA says, we can tell you whether it was safe to go into water yesterday. We have no idea whether it's safe today. That's the problem. There's absolutely no correlation, at least at this particular beach, between the Enterococcus counts measured on successive days. And this is another beach, also studied by Allie Beam. And remember, 104 is where you draw the line if you're taking single samples. This is Enterococcus. And so this is the time series of samples or results at this particular beach, Cabrillo Beach. And you can see on occasion the counts are practically off the charts. But here's a little blow up of this region around the 4th of July. And where she's showing in red here is where, or pink, is where the beach would have been closed, based on the counts the previous day. So you see, that's over 100, that's over 104, that's over 104. You would get those results 24 hours later. You'd close the beach, and the fact is the counts on the days the beaches were closed were acceptable, which underscores what Al Dufer said. We can tell you whether it was safe yesterday. We don't know whether it was safe today. And so she's published this I just mentioned. This is Environmental Science and Technology, Enterococcus concentrations, extreme variability. And then she's taken this, again, Allie Beam, to the extreme of sampling every minute or every 10 minutes, and at one place, and you can see how the counts go up and down, and again, there's the line, 104. So they're above the line or below the line, da-da-da-da-da. And this is because you have, physical processes going on. You're taking a sample about the size of a teacup. Sometimes you see them, sometimes you don't. You see, some of the counts are practically zero, and others are, well over the criterion. Now, I've been with Department of Health people go down and sample these beaches. They go down, take one sample in the morning, bring it back to the lab to get busy processing. That's it. And then that is 30 minutes. Yeah, they were sampling every minute.
But all the way to the acid.
Okay, that's 215, 245, right? Right? So they were sampling every minute. This is why you have grad students, right? And so, okay, so there's that issue. You have a lot of variability in time. You have a lot of spatial variability. And then, unfortunately, see, not all fecal indicator bacteria come from human feces. Now, do you want to be swimming in a water that was, birds pooped in? Probably not. But, you know, the assumption is that the problems are coming from human feces. So the fact is you find these same indicator organisms in places other than human feces. Birds, for example, some fish, and then what's, I think, a bigger concern is you find them in reservoirs that have nothing to do with feces, beach sands, sediments, you know, plants. They're bacteria, right? And they find a place, you know, the assumption is they live in your gut, which they do, but unfortunately you also find them living in other parts of the environment. There's nothing to do with fecal contamination. By the way, I should mention that not all of these issues are specific to feces. You do, if you're infected, you can shed some of these pathogens in your urine, but the vast majority of them are shed in feces. But you know, if you look at something like Leptospira, no, it's in the urine. That's another issue. So here's some papers that have been published about the presence of these things in other reservoirs. And I'll call your attention to this one in particular, Roger Fujioka. They found these Enterococcus in soils in Hawaii. I mean, they're natural inhabitants of the soil. And there's nothing to do with feces. But you know, the assay picks up these things. They grow under the specified conditions, and they form colonies, and you count them, and you say, oh, those are fecal indicator bacteria. They're not. Then you have the fact that some pathogens are present. I mean, these are real pathogens, and they're not human pathogens, and there's nothing to do with feces. So here they're looking at viruses, pathogenic human viruses, and they're in rivers. Now, you know, how did they get there? I don't know. But they're there, and you know, here's the various rivers that were assayed. Apparently nothing to do with fecal contamination. Now, they're human pathogens, so you don't want to be swallowing this water. On the other hand, if you're using an indicator that's going to specify fecal contamination, and you have these things there that have nothing to do with feces, that's not maybe very helpful. Okay, so you have that issue too. Just you have a wide variety of pathogens with various sources, and so here's a little summary of where these things can come from: animals, you know, feedlots, what have you, wildlife, you know, seagulls, birds, etc. Then you have soils, storm runoff. After storms, you typically have a big increase in these fecal indicator organisms. Sure, you may be flushing poop off the streets. You know, somebody's walking their dog and didn't clean up. But in Hawaii, if you have soil erosion, the soil comes along and you get the Enterococcus. Then you have human pathogens that are natural inhabitants of the environment. And so Vibrio bacteria, see, nothing to do with feces. Vibrio bacteria, a good example. And in this is a study that I was involved in, and we're looking at two types of Vibrio bacteria that definitely cause human health problems. Vibrio parahemolyticus, that's VP, and Vibrio vulnificus. Vulnificus is the flesh-eating variety. About half the people who get infected with this die. Normally, parahemolyticus just gives you a good case of gastroenteritis. And here we're looking at the concentrations of these things in oysters. And you can see these concentrations vary widely. You're looking at 6, 7 orders of magnitude here. And what we did here is look at temperature and salinity. And we correlated the incidence of these types of Vibrio bacteria with temperature and salinity, and you can explain about 70% of the variance in the data if you just know the temperature and salinity. When the temperature goes up, you tend to get a lot more of these things, and if they are happy in a certain range of salinities, and then outside of that range, no. So this is actually a nonlinear relationship, and this is just showing you the correlation between the observed and the predicted concentrations of these things. Obviously, it's straightforward to go out and measure temperature and salinity. The problem is people like to eat raw oysters. If you cook the oysters, it's not a problem because you'll kill these things. But some people really like to eat raw oysters. And then, you know, if you actually swallow the water, and if it happens to be fresh water, then you've got issues. So here's an example from Bangladesh where they get their water from surface sources, And in that case, you can come down with Vibrio Cholera, which can take your life. You basically dehydrate yourself to death. The treatment is called oral rehydration therapy, but if that, I'm running out of time here, if that doesn't work, you hook people up to an IV and drip saline solution into them. Your body will knock out the Vibrio Cholera if you don't dehydrate yourself to death first. And the simple corrective is to filter the water. That significantly reduces the likelihood of coming up with an infection. So this is what the EPA is working on, some way to cut down the time that it takes to get a result. And so they're using a real-time PCR, which just is detecting the DNA from the organism. And I pointed out here, the fact that there's DNA there doesn't mean that you have a viable organism. But if you talk to the EPA, say, we don't care, it means there were feces in the water, you know, if we have the right indicator bacterium. You have to relate these results in an epidemiological study, otherwise the EPA will completely ignore it, okay? You have to get some kind of relationship. So this is what they're looking at. Bacteroides, it's human specific, okay? So they've got a bacterium that's specific to humans, and you can see, it says a lot of your feces are actually dead Bacteroides bacteria. They're actually present in higher concentrations than the coliforms that we're using right now. And if you do your DNA right, they are host specific. Now again, you have birds, you know, pigs, chickens, whatever, but you can identify these things specific to humans. They don't survive long in that environment, so that's bad because some of these pathogens do. But here's the thing, you can look at the viruses that infect these things, and they do survive for a long time. So the assay is not going to be for the Bacteroides, it's going to be for the viruses that infect the Bacteroides. And so if you find these viruses, you can pretty much conclude that the water's been contaminated with human feces. Now, you don't know when, because they survive for a long time. But the concern is if you have things like hepatitis A in the water, they survive for a long time. And that's the kind of indicator you want. All right, why we use viral indicators? They're able to replicate in the environment, so you don't want these things growing in soils. They survive sewage treatment. And so we saw where that was an issue. Persist for a long time, like the hardier pathogens. And if you're concerned about viruses, that's what you want to look for, okay? And I'll just add, this is not where the EPA is going right now. They're still looking, they're going to look at traditional indicator bacteria just using real-time PCR. So here's a summary of the issues. This one is yet to be resolved, the temporal and spatial variability. We still have to take that on non-specificity. Bacteroides, or something like it, you know, and the viruses that infect them. This issue may be resolved by looking at viruses because they're the hardier pathogens. Rapid test, real-time PCR. This is another issue. You know, pigs, cattle, whatever. We still have to address that. And then, so you've got to do this. And these are expensive studies. Epidemiological studies are expensive. Okay, good. Thank you.